is Claire Olson, and I looked at using Rarity and Evolutionary Distinctiveness, or the degree to which species are unrelated from each other, to prioritize the protection of angiosperms. And specifically, I looked at the flowering plants of Olympic National Park. It's a relatively interesting region right in our backyard. Oops. So historically, plants have been prioritized for their rarity, their agricultural, cultural, or ornamental importance, or their value as providers of ecosystem services to animals or humans specifically. Like, they're pretty, they make shade, do good things like that. But there's a lot of plants that don't really do anything as far as we're concerned, like skunk cabbage. It sits there, it gets smelly in the spring, and it's yellow, and your kid might bring it home and make you sad with it. But like, what good is skunk cabbage? So that's the question that I really sought to answer, because there's a ton of plants, a lot of rare plants, not a lot of funding, and climate change is just going to make the demand for conservation bigger than ever, and while we would, in an ideal world, provide every species with an appropriate conservation plan, that's just not feasible given the reality of budgets and conservation work itself. So we have to make choices about what species to prioritize and when. So the question is, like, what makes a species special if it's not, good for anything, if that makes sense. So the approach John introduced me to, and then I found through ferreting amongst the literature, was phylogenetically informed conservation. So prioritizing species for conservation based on how much they can, like, what share of evolutionary history they present within their genera. So evolutionary distinctiveness is the most popular of these metrics because speciation does not occur evenly in all places at all times. And this method kind of averages speciation rates among like all of the species in your phylogeny and makes them comparable to each other. And it's calculated by taking the sum of all of the branch lengths, represented as millions of years before the present, within a group, and then dividing that number across the group's members. So each species has an evolutionary, so in this hypothetical phylogeny, evolutionary distinctiveness scores are listed on the right, and the numbers above the nodes represent the branch length, and the numbers below the nodes represent the members, like a kind of the number of members in each branch of that. After you run your calculation, the species with the greatest evolutionary distinctiveness score are the most unrelated to other species within their group. And so in this example, species F and G would be prioritized for conservation because they have the highest ED scores, if that makes sense. So it's been 2.75 million years since they diverged from their next closest relative, if that makes sense. I chose to work with Olympic National Park because of its, well, the Olympic Peninsula's status as a refugium during the last glacial maximum. Refugia are areas where there is a combination of habitats or environmental factors and the organisms within those areas that together lessen the impact of disturbance, which is pretty interesting. And the factors that made the Olympic Peninsula a refugium during the last glacial maximum are set to make it a relatively safe harbor for species during our current period of warming. So it's an area that has been long prioritized for conservation, but has had that attention really heavily focused on the critically rare species in the park, like spotted owls or the plant taxa at the alpine zone in the park, which we're losing because of climate change. Things get warmer and you run out of mountain pretty fast. So there's not been a lot of work on any other plant species, and figuring out how to prioritize them would be pretty useful, I would say. In order to calculate evolutionary distinctiveness, you have to have a phylogeny. This, where the branch lengths represent time since divergence for taxa, and this turned out to be, acquiring a phylogeny turned out to be quite a strange trip. The phylogeny of plants as a whole is not particularly well resolved, but the evolutionary relationships of flowering plants or angiosperms, but the single tree that was calculated for angiosperms is only available through a server at Harvard that is available through a C program called Phylocom or a website called the Phylomatic, and only accepts text files of your taxa in a very long single line list, if that makes sense. But otherwise, it acts just like the R packages, ape, caper, that perform these functions as well. And so after a lot of searching, I was able to use the Phylomatic to access my phylogeny of choice, trim it to just the species within Olympic National Park, and then perform the evolutionary distinctiveness calculation. But after running my list for the first time, I received just a huge number of taxa not returned, which was quite strange because I knew that based on the publication for this phylogeny that all of my species should be in there. And this is where taxonomy came into play, because since this phylogeny was published, and even before then, so whenever this species list for the park was compiled, many species have undergone name changes or even genus changes, family changes. And because the Phylomatic was making returns from the phylogeny based on, family, genus, species name, I was missing a lot of results because I had the wrong family, genus, or species name. So I spent a week and a half updating all of my names to those used in the phylogeny, which, because it was built in 2014, did not match contemporary 2021 nomenclature, which was an adventure in and of itself. But I was able to trim my list from 1,300 species to 871, which is more manageable. There you go. That is the phylogenetic relationships of the angiosperms of Olympic National Park. As you can see, it's a pretty diverse group of lineages, which is really nice to see. Like, we could lose a lot out here at these little tips before we'd really start losing big chunks of this phylogenetic tree. Just, you take happy climate change news where you can, right? So to spare you from looking at a 37-page table, I have chosen to represent my results relative to what was happening in geologic time. So out here at about 180 million years is kind of the early Jurassic period. It was very warm and wet. We only had one taxon persisting that long, which makes a lot of sense. That was a very, very long time ago. And this is wild ginger out here at the very furthest end. And then as you get closer to the present, at about 130 million years, we're coming out of the Jurassic into the Cretaceous. It's getting a little bit drier, but it's still very wet and tropical around the globe. And it stayed that way until the Eocene. And as you can, so around 100 million years is where commonly people start talking about like the rise of angiosperms. That makes sense, the rise of flowering plants, and these results match that. So overall, I didn't find any particularly novel patterns in the abundance or incidence of speciation events in my group, because they matched major extinctions and radiations of plants relative to carbon dioxide in the atmosphere. Like at the border of the Eocene and Oligocene, there was a major extinction event, and you see that subsequent dip in here, which makes sense. So each of these big dips are major extinction events after the rise of angiosperms, which is interesting, but not particularly noteworthy in and of itself. Overall, there was a very wide diversity of evolutionary distinctiveness scores, but most of these species had scores between 70 and 45 million years of age, which again reflects that major radiation event as grasslands spread after the Eocene. Overall, old things you would look at out in your yard and say, oh, that's a strange old lineage, that's a strange old plant. are strange old plants. Wild ginger up here at the top, Asarum caudatum, is a very strange old plant. Brasenia is a weird old lily. It's been hanging out a long time. It makes sense that it's has a very high divergence time from its nearest neighbor. And there were some surprises, like Carex is a genus of sedges, and sedges have been around quite a long time. They came around before grasses. And that genus had some members in the 70 million year old group, but also some very new members here down at the very bottom of my rankings at just under 3 million years of age since they appeared. So overall, it's just reinforcing the fact that Olympic National Park is home to a very diverse community of flowering plants, and it would be really interesting to see what's going on with non-flowering plants as well. I also, so I wanted to see how well prioritizing plants for conservation based on their rarity matched up with prioritizing plants for their phylogenetic diversity, if that makes sense. And this was not, I feel this would have been more informative if I had chosen the resolution of the whole state of Washington rather than such a small area. With only 40 species in my sample, the results were very homogeneous. The average threat ranking was 2, and most species were like they'd all diverged around 45 to 60 million years ago. So this is not a particularly interesting or informative result, other than saying that at least within the park at this small scale, rarity was not a good proxy for how distinctive species are within their genuses. You can see that here. So at either end of the scale, the very oldest species and the very youngest species were noteworthy and had differences in their ready scores, but overall, this was a very homogeneous group when I tried to weight evolutionary distinctiveness by threat. So in conclusion, evolutionary distinctiveness seems like a promising and useful means for identifying phylogenetically important species and therefore basing the value of unimportant plants, so to speak, in their contribution to biological diversity and their ability to harbor genes that will allow future speciation instead of prioritizing new species that do not represent the same share of genetic diversity. And based on my relatively small sample, I would say that prioritizing rarity alone is insufficient to prevent the significant loss of evolutionary history through climate change, especially since plants are underfunded overall. Better to focus on more distinctive species. So, thank you for your time.